Bismillah ar-Rahman ar-Rahim. This is the third lecture of embryology. We're going to talk about the embryogenesis during the third week of development. As we mentioned in the previous lecture, the cleavage process includes dividing of the fertilized ovum into 2 cells, 4, 8, until it reaches the stage of morula formation. This morula contains 16 cells. Each one is named the blastomere. It ends with the implantation of the embryo inside the wall of the uterus or the endometrium. Morphogenesis occurs in this stage. Morphogenesis is the process by which cells occupy their appropriate locations. They involve gastrulation. It means the movement of cells from the blastula surface to the interior of the embryo. Also, the organogenesis is the formation of organs. So the gastrulation, it means rearrangement of the cells of the blastula into the 3-layered embryo called the gastrula. The blastula, as you know from the previous lecture, it means the blastocyst. It contains 2 parts, the outer cell layer, which is the trophoblast, and the inner cell layer, which is the embryoblast. It will change from blastula into gastrula. The blastula, the embryo, is composed of 2 layers, the epiblast and hypoblast. Then, after gastrulation, the embryo will have 3 cell layers. 3 layers produced by gastrulations are named the embryonic germ layers. The ectoderm, endoderm, and mesoderm, which lies between both layers. Each germ layer contributes to specific structures. Now, gastrulation is the most characteristic event occurring during the 3rd week of gestation. It's defined as the process that establishes all 3 germ layers, as we mentioned, the ectoderm, mesoderm, and endoderm in the embryo. The innermost layer is the endoderm, the outermost layer is the ectoderm, and in between lies the mesoderm. Gastrulation begins with the formation of the primitive streak. This is the primitive streak. It's a very important structure. It gives a landmark for the beginning of the gastrulation process. It's clearly visible as a narrow groove, slightly bulging regions on the either side of the caudal end of the embryonic disc. This represents the embryonic disc. This is the caudal end, and here is the cranial end. The primitive streak is seen here at the caudal end of the developing embryo. This part of the primitive streak is named the primitive node. It consists of a slightly elevated area and a small primitive pit in the middle. So this represents the primitive streak. It's a very important event that occurs just before gastrulation. This is a primitive groove, and this is a primitive pit in the middle, and the primitive node, which is a slightly bulging area. This is the cloacal membrane, or at the caudal end of the embryo, and this is the cranial end of the embryo. The primitive streak consists of 3 parts: the primitive groove, the primitive node, and the primitive pit. Appearance of this primitive streak or this structure indicates the craniocaudal axis. We can determine the cranial end and the caudal end of the embryo. Also, we can determine the dorsal part and the ventral surfaces of the embryo. Also, right and left sides could be identified at this stage. By the end of the 4th week, the primitive streak shows regressive changes, shrinks, and disappears. This will later on at the end of the 4th week will disappear. This slide shows the bilaminar germ disk where the primitive streak appears here. So this tells us that this is the caudal end of the embryo, this is the cranial end, and this is also the dorsal side, and here there will be the ventral side. This is the primitive streak. Here again, this is the bilaminar germ disk, amniotic cavity, yolk sac, and surrounded by the trophoblast and the syncytial trophoblast. Here it is implanted in the endometrium of the uterus. About the mechanism of gastrulation, cells of the epiblast migrate toward the primitive streak, and when they arrive the region of the streak, they become flask-shaped. They detach from the epiblast, slip beneath this streak. This inward movement of these cells is known as the invagination. Once the cells have invaginated, some displace the hypoblast cells. They create the embryonic endoderm. Others come to lie between the epiblast and the newly created endoderm to form the third layer, which is the mesoderm. The rest of the cells that remain in the epiblast, they form the ectoderm. Thus, the epiblast, through the process of gastrulation, is the source of all the germ layers, and cells in these layers will give rise to all of the tissues and organs in the embryo. Here, this is the primitive streak, and this is the Hence's node or the primitive node. These arrows point to the direction of the migrating cells. This is the epiblast. Cells around this structure, or they are close to this structure, they start to detach from this epiblast and they will migrate in this direction, entering the primitive streak. Here you can see that these cells, after arriving in the area between the epiblast and hypoblast, they will displace some of the cells of the hypoblast, giving the endoderm. The rest of the cells remaining in between the epiblast and the hypoblast, they will give the mesoderm. The remaining cells on the epiblast, they will give the ectoderm. So at the beginning, the ectoderm is named the epiblast. After this stage, it will change into ectoderm. This is the direction of the migrating cells. These cells represent the mesoderm. You can see the direction of the arrows indicating the direction of migration of the cells from the epiblast, entering this structure, and then will end in the area between the epiblast and the hypoblast. Now it's named the ectoderm, endoderm, and in the middle they are the mesoderm. Another figure shows this mechanism. Here you can see the primitive streak through which the cells of the epiblast migrate. They will migrate in the lateral direction on both sides, displacing some of the hypoblast cells, forming the endoderm. Other cells remain in this area, will give the mesoderm. This part of the mesoderm is known as the intra-embryonic mesoderm. It will be in contact with the other part of the mesoderm, which is named the extra-embryonic mesoderm. This figure shows simply the process of gastrulation in mammals. You remember the blastula, which is composed of a trophoblast, and the inner cell mass will give the embryoblast. Here the amniotic cavity is very clearly seen above the epiblast, and this is the yolk sac. At this stage, this layer shows it's named as the extra-embryonic mesoderm. here at the 3rd week, the gastrulation is beginning, forming the 3rd layer, which is the mesoderm. More and more cells move between the epiblast and hypoblast. They begin to spread laterally and cranially. Gradually, they migrate beyond the margin of the disc and establish contact with the extraembryonic mesoderm. In the cephalic region, or the cranial direction, they passed on each side of the precorder plate. This plate itself forms between the tip of the notochord and the oropharyngeal membrane. We'll talk about them later. And it's derived from some of the first cells that migrate through the node in the midline and move in the cephalic region. The oropharyngeal membrane, it is seen at the cranial end of the embryonic disc, or the head region, consists of a small region of tightly adherent ectoderm and endoderm. There will be no mesoderm in between them. Represents the opening of the future mouth. This is again the embryonic disc. Here is the primitive streak at the posterior part of the dorsal side of the embryo. It's a primitive node and pit. The migration of the cell occurs through this groove, and they will move in the lateral direction and in the cranial direction. This is again the primitive node, and this is the primitive streak, showing the direction of the movement of these cells from the epiblast, displacing some of the hypoblast cells, giving the endodermal layer, and in between lies the 3rd layer, which is the mesoderm. Now, each of these 3 layers that appeared after gastrulation give different derivatives. The ectoderm gives epidermis of the skin and its derivatives, nervous and sensory system, pituitary gland and adrenal medulla, jaws and teeth, and germ cells. The mesoderm gives skeletal muscular systems, circulatory and lymphatic system, excretory reproductive system, dermis of the skin, adrenal cortex. The endoderm gives the epithelial lining of digestive tract and associated organs, epithelial lining of respiratory and excretory and reproductive tracts and ducts. Lastly, the thymus, thyroid, and parathyroid glands. Thank you for your listening.